Welcome today to Amerit's Oncology webinar. Today, we'll be providing an informative overview of reader variability in oncology clinical trials. My name is Bria Augustino, and I'm an Oncology Business Development Associate, where I support clinical research by identifying sponsor needs, building relationships, and connecting them with the right solutions. I am pleased to be joined by Will Butte, our presenter today. Will's role as an oncology proposal development specialist focuses on supporting oncology clinical studies through scientific and market research by drafting proposals, white papers, and clinical trial imaging protocols. Will holds a BA in biology from Cornell and a master's in biomedical science from Rutgers, with 3+ years of graduate research on hematologic malignancies at Rutgers.
Thank you, Bria, and thank you all for joining us today. I'm glad to have the chance to talk with you about reader variability in oncology clinical trials, what it is, why it matters, and what we can actually do about it. This is a topic that sits at the very center of what an imaging core lab does, and it has real consequences for how trials are designed, run, and ultimately interpreted. Before we get into the details, let me give you a quick roadmap for today. I'll begin with some background on what reader variability actually is, how blinded independent central review works, and why this issue matters so much for trial integrity. From there, we'll look at how common reader variability really is, using data pooled across large multi-trial data sets. Then we'll break the problem down, first by separating inter-reader from intra-reader agreement, and then by walking through the leading causes of discordance. Next, we'll examine how variability differs across response frameworks and across cancer types, because the patterns are not uniform. And finally, we'll look at the part that matters most in practice, how we mitigate this variability and where the field is heading, including the emerging role of artificial intelligence. So let's start with some background by defining reader variability, situating it within the blinded independent central review process, and explaining why it's such an important issue for the integrity of a clinical trial. So in oncology trials, tumor response assessment is the foundation for determining whether a treatment works. Almost every imaging-based efficacy endpoint ultimately comes down to a radiologist looking at a scan and making a call. Is the tumor growing or shrinking? Are new lesions appearing compared to previous scans of the patient? Basically, is the treatment working, or is the disease still progressing or remaining stable? So to keep that process consistent, we use standardized response criteria like RECIST, RANO, and others. The whole point of these criteria is to standardize how radiologists measure and categorize tumor change so that a trial's result reflects the treatment and not the particular reviewer who happened to read the images. Reader variability is the disagreement that remains between qualified readers even when they are evaluating the same images under the same criteria. It's what's left over after we've standardized everything else that we can. And the key point I want to plant early is this: reader variability is a persistent expected phenomenon. It shows up across response criteria, across cancer types, and across imaging modalities. It is not simply a sign that a reader has made a mistake, which is a common misconception in the clinical trial industry. And that distinction is going to come up again and again throughout this presentation. So how do we manage this in a trial setting? The answer is blinded independent central review, often abbreviated to BICR. To minimize investigator bias, the FDA recommends blinded independent central review for pivotal oncology imaging trials. Rather than relying only on the local site read, the images are sent to an independent core lab for centralized evaluation. The way it typically works is that two independent readers evaluate the same images, each blinded to treatment assignment and to each other's reads, in order to minimize bias from any possible source. When those two readers disagree, a third reader, called the adjudicator, steps in to resolve the discordance. And that mechanism gives us a very useful practical metric. The adjudication rate, which is simply the share of patient scans that required a third reader. Because it directly counts how often two qualified readers disagreed, the adjudication rate is the most common real-world measure of reader variability, and you'll see it referenced throughout today's talk. Sometimes the adjudication rate is referred to as discordance rate or disagreement rate, and it can also be measured as the inverse, like agreement rate or concordance rate, but it all essentially means and measures the same thing, which is reader variability. And here's the important part. BICR is a safeguard and a good one, but even with it in place, meaningful disagreement between readers remains common. Understanding how common and why is exactly what this review examines. So before we quantify the problem, let's be clear about why this is important. Reader variability isn't just an academic curiosity. It has direct practical consequences for a trial. The first of those is endpoint integrity. Endpoints like progression-free survival and objective response rate depend directly on when and whether a reader calls progression of the disease. If two readers disagree on that call, they can produce two different endpoint values for the same patient. Second is statistical power. Variability in response assessment adds noise, and that noise can reduce the power to detect a true treatment effect. So it blurs the difference between trial arms that the study is designed to measure. The third is cost and timeline. Every adjudication adds a third read, which requires a third radiologist to be brought on, and at scale, that adds real cost and can extend the duration of a trial. And fourth is regulatory confidence. Consistent, reproducible reads strengthen the credibility of a submission, while inconsistent ones can invite questions and doubts. The through line here is that understanding the root causes of reader variability is the first step toward mitigating them, and doing that helps improve trial efficiency, lowers costs, and ultimately improves patient outcomes in the fight against cancer. So now let's put some numbers to it. In this next section, I want to look at adjudication and discordance rates across some large multi-trial data sets, because the scale of this is often surprising, even to people who work in the field. The headline here is that across BICR-based solid tumor trials, disagreement happens far more often than intuition would suggest. If you take an average across BICR solid tumor trials, the adjudication rate sits at roughly 40%, and in some indications, it climbs as high as 65%. So in some studies, two qualified independent experts can disagree on nearly 2 out of every 3 patients. That's not just from one small study either. In a landmark paper for the reader variability question in oncology clinical trials, Ford and colleagues looked across 79 trials and more than 23,000 patients and found a 42% patient-level discrepancy rate overall. Beaumont and colleagues, looking specifically at 6 lung cancer trials, found an even higher patient-level discrepancy rate of 59%. So whichever data set you look at, the message tends to be the same. Reader disagreement is not a rare edge case. It's a routine feature of how these trials actually run. Now, this table pulls together the key evidence in one place so you can see how consistent the pattern is across independent research groups. Like I said before, Ford and colleagues in 2016 reported a 42% adjudication rate across 79 trials and over 23,000 patients, with a range from 21% in renal cell carcinoma up to 55% in ovarian cancer. That range is worth noting. We'll come back later to why cancer type matters so much for reader variability. Beaumont and colleagues in 2021 reported 34% discordance at the time point level and 59% at the patient level across 6 lung cancer trials. INSE and colleagues found 39% discordance specifically on the date of progressive disease across 5 lung cancer trials. Borodale and colleagues in a very large data set of 40 trials and more than 12,000 patients found 23% disagreement on best overall response and 31% on the date of progression. And Gorsey and colleagues in a smaller study in pediatric diffuse intrinsic pontine glioma using RAPNO criteria found readers concordant in 68% of paired reads, which they had specified had 25% minor and 7% major discordance. So across different cancers, different criteria, and different research groups, the story is remarkably consistent, which is to say that, at least for now, reader variability remains a structural feature of imaging-based response assessment, but fortunately, it is one that can be mitigated and controlled, which we will touch on later. Now, I want to be balanced here because it would be easy to walk away from that last slide a little overly alarmed. High disagreement at the individual time point level does not necessarily overturn a trial's overall conclusion. A good illustration of that comes from Tang and colleagues in 2025. In a meta-analysis of 70 hematology-oncology trials conducted over a decade, they found a pooled hazard ratio of 0.96 with a tight confidence interval. This indicates a strong agreement between investigator and central review at the level of the treatment effect. Even more striking was that across the 33 trials where progression-free survival was the primary endpoint, investigators and BICR reached perfect concordance on whether the result was statistically significant. In other words, the trials landed on the same answer. Part of the reason for this is that in hematology, assessment is often multimodal, incorporating labs and histopathology, and those more objective quantitative measurements reduce reliance on subjective imaging interpretation. And also, aside from lymphomas, where solid tumors can form in the lymph nodes or organs, leukemias are essentially entirely assessed as abnormal cells in the bloodstream and bone marrow, so there aren't really any solid tumors or lesions that need to be counted or measured, which is a significant source of variability in solid tumor imaging. So the overall takeaway is a measured one. Variability is real, and it is absolutely worth controlling, but well-designed trials built on objective endpoints remain robust, and certain types of cancers, by their nature, will be less ambiguous in their presentations. The goal, again, is to manage the variability, not to fear it. So to manage variability, we first have to understand where it comes from, and that starts with an important distinction between two different types of agreement, which tells us a great deal about the dominant source of the problem. There are really two kinds of reproducibility or variability that we care about. The first is intra-reader agreement. That's within a single reader. It asks if the same reader evaluates the same images at two different time points, do the readers agree with themselves? Essentially, is a single reader self-consistent? The second is inter-reader agreement. That's between readers. It asks if two different readers evaluate the same images, do they agree with each other? So do two qualified experts reach the same conclusion? And here's the general finding across the literature, and it's what you might intuitively expect. Intra-reader agreement is consistently higher than inter-reader agreement, so readers tend to agree with themselves more than they agree with each other. That leads to an important implication. If the biggest gap is between readers rather than within them, then the multi-reader paradigm itself, the fact that we use more than one reader, is the dominant source of discordance. So it's an inherent part of BICR. However, it's not primarily that individual readers are inconsistent or making mistakes. Very often, two experienced expert readers can simply have medically justifiable differences in how they interpret very ambiguous disease presentations. This chart makes that concrete using ovarian cancer data from Krasovitsky and colleagues. For each category, the purple bar is intra-reader agreement, and the green bar is inter-reader agreement. What you see generally is that intra-reader agreement usually exceeds inter-reader agreement at most levels, except for lesion selection, peritoneal in this case. Overall lesion selection, overall lesion measurement, and lymph node measurement. The gap is especially wide for the harder tasks like lymph node assessment, where disease is more diffuse and harder to define. In fact, in this cohort, inter-reader differences accounted for roughly 85% of the total measurement variability. So the between-reader component isn't just larger, it's the dominant source of variability by far. This is an important insight operationally because it tells us where to focus our mitigation efforts, which is on the sources of disagreement between readers. And of course, this isn't unique to one particular type of cancer, like the ovarian cancer example from the previous slide. The same pattern shows up across very different indications and modalities. In ovarian cancer, under RECIST 1.1, as we just saw, inter-reader measurement differences accounted for about 85% of total measurement variability. In prostate cancer, using PSMA PET-CT, three readers assessing 125 scans were found to agree on 77% of cases between readers, but agreement rose to 90 to 95% within a reader on rereads. So the same images, but much higher agreement when it's the same person reading twice. And then in lower grade glioma under RANO response criteria, intra-reader reproducibility was moderate to excellent, while inter-reader reproducibility ranged from poor to moderate. So we know disagreement is common, and we know it's mostly a between-reader phenomenon. The next natural question is, what specifically are the readers disagreeing about? Let's look at the leading drivers, and importantly, at how much of this is justifiable difference versus true reader error. This chart, using breast cancer data from Borodale and colleagues, breaks down the share of discordant cases by their root cause. The single largest driver is lesion selection differences at 37%, where readers are choosing different lesions to measure. Close behind that is new lesion perception at 30%, where one reader is seeing a new lesion that the other doesn't. Then there's non-target progression at 13%, image quality or missing data at 11%, and finally lesion measurement differences at 9%. And that last number often surprises people. We tend to assume that variability is about millimeters, about readers measuring the same lesion slightly differently. But pure measurement disagreement is actually the smallest contributor. The real drivers are about which lesions readers look at and whether they perceive a new disease at all. Essentially, quantitative measurements tend to produce less variability than qualitative ones, the more qualitative ones generally involving differences in perception rather than direct quantified measurement. And that concept that new lesion detection is consistently the single most frequent cause of discordance across essentially every response framework is important enough that it deserves its own specific discussion. So new lesion detection comes up again and again as the top driver of discordance. Across 5 lung cancer trials analyzed by INSC and colleagues, discordant progression calls broke down as follows. So 41% were attributable to new lesion detection, 33% to target lesion diameter increases, and 12% to non-target lesion progression. A separate analysis of a small lung cancer trial went even further, attributing 54% of all adjudications to differences in reporting new lesions. So why is this so difficult? It's mostly because new lesion detection is fundamentally a perception task. It comes down to whether one reader sees a finding that the other simply does not. And unlike a measurement, which we can govern with a numeric threshold, perception is very difficult to standardize with a rule. You can't really write a percentage cutoff for did you notice this? And that's a big reason why this category resists the usual standardization tools and why it remains such a persistent source of disagreement. The other major driver is baseline lesion selection, and this one can be particularly problematic because the lesions a reader chooses at baseline shape every subsequent assessment for that patient, and early divergence propagates through the entire follow-up. Like if you think about a math exam, if you get the first part of a multi-question problem incorrect, all your answers to subsequent questions will be incorrect as well. And the data bear this out across 5 lung cancer trials with more than 1,700 patients. Readers targeted at least one lesion in a different organ in 44% of patients, most often in the lymph nodes at 20% and in bone at 12%. And these weren't random differences. Reader-specific selection patterns, things like the mean sum of the diameters they choose and where in the body they focused, were systematically different with high statistical significance. That finding points to consistent behavioral differences between readers, almost like a reading style. Perhaps the most striking single statistic, in one breast cancer study, 2 experienced radiologists were found to have selected the same target lesions only 28% of the time. So that's the same patient, the same images, the same criteria, and the readers agreed on which lesions to follow barely a quarter of the time. This brings us to what I think is the most important conceptual point on this topic. It would be easy to assume that all of this disagreement somehow represents error or mistake, that someone is getting it wrong, but that's not really what the data show. Most discordance is not a mistake. It simply reflects genuine differences in medical judgment applied to ambiguous images by very experienced and skilled radiologists. Roughly 75% of discordant cases reflect medically justifiable perception differences, not reader error. These are cases where 2 experts, both reading correctly and in good faith, simply interpret an ambiguous finding differently. About 49% of discrepancies were a timing difference, not a disagreement about whether progression happened, but about when. And in 80% of those, it was just a 1 cycle offset. So one reader called it at this scan, and then the other called it at the next. And there's a nice illustration of how confirmation helps. A progression call supported by multiple RECIST components at once was found to carry a positive predictive value of 0.89 in one study versus just 0.59 when it rests on the sum of diameters alone. So more corroborating signals produce more reliable calls. And the reframing here is crucial. We are largely not trying to fix bad readers. That isn't really a thing that happens very often in reality. What we're trying to do is manage legitimate expert disagreement about images that are genuinely and often unavoidably difficult to read for a multitude of reasons. That being said, I don't want to overstate it and imply that nothing can be improved. A systematic review of blinded double reads draws a useful line between the 2 major categories of discordance here. The first is medically justifiable differences, the legitimate differences of expert opinion on genuinely ambiguous findings that we just discussed, which is difficult to mitigate. However, the second category is mostly avoidable errors, things like noncompliance with the reading charter, missed lesions, or inconsistent window and level settings on the images. The contributing factors here include reader expertise, image quality, and whether the reader has access to the relevant contextual information. And this distinction really matters because only this avoidable portion can be reliably engineered away through better training and better charter design. The rest is mostly inherent to BICR. The natural characteristics of certain cancers' presentations make many of them inherently more difficult to interpret than others, which is what we'll cover next. So now let's look at the response criteria we use and the underlying disease biology and how they shape the amount of discordance we see. Because as those earlier ranges hinted, variability is far from uniform. So let's start in the frameworks themselves. This table covers 3 of the most common criteria in solid tumors. RECIST 1.1, which is the workhorse of solid tumor trials, carries a roughly 40% adjudication rate overall. Its main challenges are new lesion detection and its relatively permissive lesion selection rules, which give readers latitude that leads to the selection differences we saw earlier. RECIST, used in the immunotherapy setting, shows substantial to almost perfect inter-reader agreement and moderate to excellent inter-reader agreement, with a kappa statistic around 0.69 for confirmed progression. Its central challenge is pseudoprogression, but its built-in confirmation step actually helps improve concordance. And just as a short aside, since it is a common known phenomenon that often directly impacts variability in some response frameworks, pseudoprogression is when a tumor can appear to grow after treatment, when in reality the enlargement is simply due to treatment-induced inflammation or an influx of immune cells rather than true disease progression. And PERSIST, which is PET-based, that's positron emission tomography, achieves better inter-reader agreement than qualitative visual PET reads alone, with high reproducibility on its key quantitative metric. Its challenges are defining the hottest lesion and harmonizing across the different scanners used across the trial. The theme here to notice is that the more quantitative and rule-based the framework, the better the agreement tends to be. This second table covers frameworks for some of the more challenging disease settings. The RANO family, or Response Assessment in Neuro-Oncology, which includes RANO, RANO 2.0, and RAPNO for pediatrics, shows poor to moderate inter-reader reproducibility and around 68% agreement in DIPG. The challenges in these areas are pseudoprogression plus diffuse T2 flare margins that are hard to delineate, which is why volumetric 3D assessment tends to beat 2D. For mRECIST for hepatocellular carcinoma, actually shows strong agreement with a Goetz AC1 statistic of 0.8 in the transarterial chemoembolization setting, though it depends on carefully delineating viable enhancing tumors as well as taking into account the necrosis of tumor cells. Lugano criteria in lymphoma generally uses the Deauville scale, which is a 5-point visual scale of scans used to evaluate how well a patient's lymphoma is responding to treatment, where level 3 in the scale represents the borderline score between positive and negative response. For reader variability in lymphoma, there is fair to moderate agreement on the full 5-point scale, which improves when it's collapsed to the binary split along the level 3 borderline. The major challenge here is the borderline Deauville 3 score and also residual masses. And finally, the non-lymphoma hematologic malignancies show that a pooled hazard ratio of 0.96 and show near perfect concordance on progression-free survival significance, precisely because multimodal assessment reduces reliance on subjective imaging. So the lesson across these frameworks tends to be pretty clear, which is that the framework and the biology together determine how much variability we should expect, which is an important consideration when designing a new trial based around a particular response criteria framework or cancer type. This chart ranks adjudication rates by cancer type, and the spread is dramatic. And importantly, it's driven by disease biology, not by reader skill. At the low end, renal cell carcinoma sits around 21% and prostate cancer around 26%. But at the high end, ovarian cancer is around 55% and basal cell carcinoma reaches roughly 69%. That's more than a threefold difference from top to bottom. But the pattern here is intuitive once you notice it. Agreement is highest where lesions are well-defined or where objective data supplements the imaging, and lowest where disease is diffuse or poorly marginated, and therefore genuinely harder to measure. So when we benchmark a trial's adjudication rate, we always have to do it in the context of the specific disease indication. For example, a 50% rate might be a red flag in renal cell carcinoma, but it would be entirely expected in ovarian cancer. So why do we see so much of a spread here? At the low variability end, renal cell carcinoma at 21%, prostate around 26%, you tend to have well-defined lesions or very structured criteria and objective endpoints, like the prostate cancer working group criteria in prostate cancer. At the high variability end, ovarian cancer around 55%, basal cell around 69%, you have diffuse or multifocal disease, peritoneal spread, and disease that is simply poorly measurable by RECIST in the first place. And underneath all of this, four key factors tend to interact. First is disease biology, which is how well defined and measurable the lesions are. Then there's framework complexity, which is how much qualitative judgment the criteria demand. Imaging modality, so CT versus MRI versus PET, etc. And then treatment type. So, for example, immunotherapy and targeted agents can produce atypical response patterns like pseudoprogression that we discussed before that can make assessment harder. Understanding all of these factors lets us anticipate variability before a trial starts, and that's what makes it manageable. Now we'll go over some of the ways that clinical trials can practically address the reader variability problem, what we do about it, and where the field is heading. I'll cover mitigation strategies like training, charters, and quantitative methods, and also touch on the emerging role of artificial intelligence as it pertains to this topic. So the mitigation toolkit really tends to fall into two buckets. There's a people side and a process side, and then there's the separate measurement and data side. So on the process and people side, it starts with reader qualification and training, indication-specific certification, and protocol-specific training for each trial so that readers know exactly how that study defines things. Group case review of challenging images is enormously valuable for building consensus. Less experienced readers in particular benefit tremendously from working alongside very experienced readers on specific disease indications and read types, so they can become familiar with some of the very specific idiosyncrasies found only in certain disease presentations. We can also use performance monitoring. It has been suggested that readers with outlier endorsement rates below 25% may need to be flagged for retraining. And underpinning all of it, standardized BICR charters that define criteria modifications, dating conventions, and missing data rules up front. A specific comprehensive charter can drastically reduce the number and significance of instances of reader variability that could foreseeably arise during a trial. On the measurement and data side, wherever possible, we should try to favor quantitative over qualitative assessment. For example, semi-quantitative persists criteria outperforms a purely visual PET read, and volumetric 3D measurement shows better inter-reader agreement than 2D in gliomas. Multi-component confirmation helps too. Requiring regression on two or more criteria was found to raise that positive predictive value we discussed before from 0.59 to 0.89. And clinical trial data transfer, which is giving readers the relevant clinical context beforehand, reduces the information asymmetry that drives some discordance between readers with different backgrounds or levels of experience. And none of these are a silver bullet on their own, but together they can meaningfully reduce the avoidable portion of reader variability. And one thing I want to emphasize is that variability can be tracked prospectively during a trial, not just audited after the fact. That's a shift from treating it as a post hoc problem to managing it in real time. For example, a useful warning signal is a data progression discrepancy rate above 50% or so once you've evaluated more than 50 patients. That could be a trigger to step back and review what's happening or what's causing that. Of course, this also depends upon the variability factors we discussed earlier, which will cause variability rates between disease indications to differ according to disease biology, framework complexity, imaging modality, and treatment type. There's also a helpful timing benchmark because, as we saw earlier, roughly 80% of timing discordances turn out to be just a single cycle offset. A one cycle offset rate itself becomes a useful quality metric to monitor. And on the technical side, scanner harmonization matters a great deal. Standards like EANM and the EARL reduce the standardized uptake value variability that affects persist and Deauville scoring. Um, and the RANO 2.0 criteria recommends the standardized brain tumor imaging protocol for harmonization. These upfront harmonization steps can help prevent variability from ever entering the data in the first place. Finally, let's look ahead to some newer developments in this area. Automated quantitative response assessment holds some real promise here, precisely because it can reduce the manual perception-driven steps that we've shown can cause the most variability. On the RECIST side, a 2026 systematic review found that artificial intelligence methods can now assign RECIST response categories directly, either from the images themselves or from radiology reports, validated against reference standards. And for brain tumors, automated longitudinal RANO tools, such as those emerging from the brain tumor segmentation, or what's called the BRATS challenge, from which AI researchers and clinicians have been competing to develop automated deep learning algorithms for brain tumor analysis and tumor response evaluation. That helps to aim to deliver objective, reproducible progression calls. I do want to be realistic about where this stands, though. The credible role for AI today is as a consistent second or third reader, or as a real-time assistant to a human reader, not as a fully autonomous single reader making unsupervised calls. This is in keeping with the history of most new technologies. AI is a powerful new tool, but one that must be used in concert with human experts, not used to replace them. And of course, there are real open challenges before we even get further than that. External validation on independent data, generalization across different scanners and acquisition protocols, and of course, regulatory acceptance and standardization of how, when, where, and what AI can be used for in oncology clinical trials. So, yes, this is a very promising direction and one that we should be watching closely. But for the foreseeable future, for a multitude of reasons, AI should only complement skilled readers rather than replace them. So here are the key takeaways. First, reader variability is a consistent cross-indication challenge in oncology BICR, averaging around 40% adjudication in RECIST-based solid tumor trials. Again, it's the norm, not the exception, and it's an expected inherent part of BICR. Second, and most importantly, most of that discordance reflects genuine medical ambiguity, not error, and it concentrates in specific places: new lesion detection, peritoneal and nodal assessment, and baseline lesion selection. Third is inter-reader disagreement, which consistently exceeds intra-reader agreement, which tells us that the multi-reader paradigm itself is the dominant source of variability and points us toward where to focus our mitigation efforts. Fourth, variability is highest in diffuse or metabolically ambiguous disease, where reader perception plays a bigger role, such as in an ovarian, glioma, lymphoma, hepatocellular carcinoma, and lowest, where objective and quantitative data supplements the imaging. Fifth, and the good news, semi-quantitative criteria, confirmation steps, standardized charters, reader training, and emerging AI tools all measurably reduce the avoidable portion of variability. And finally, the reassuring bottom line: well-designed trials built on objective endpoints and strong charters remain statistically robust despite time point level disagreement. Um, Variability is manageable, and managing it well is exactly what a strong imaging partner brings to the table. Okay, so for those who might want to go deeper, here are the primary sources behind today's discussion. This information, along with the full reference list, will be available on our website and YouTube page after the webinar. And again, this webinar doesn't quite represent a completely comprehensive review of the entire scientific literature on the reader variability and oncology clinical trials topic. But hopefully this presentation was sufficiently in-depth, informative, and helpful enough to you to give you a good understanding of this problem and what we can do to continue to be aware of it and address it moving forward.
Thank you, Will, so much for providing those insights. We're switching gears a little bit as a reminder to who Merit is. Merit has served biotech and pharmaceutical companies in their clinical endpoint analysis for over 14 years with a team of medical professionals and seasoned operational support staff. Our company originally started in ophthalmology, but has expanded to include oncology and respiratory therapeutic areas. We are based out of Madison, Wisconsin, in the United States, but our company has a global reach. We have offices in China, Australia, and even Canada. We also have team members in reading centers around the globe, making it possible to support sites and sponsors from a variety of locations. Our approach is to focus on three key areas. Our software system, Excelsior, can handle all aspects of an imaging core lab in one transparent system. From acquiring images from a study site to performing an independent read and transferring data to a sponsor, Excelsior can manage and track images every step of the way. Merit has a large network of over 300 radiologists and oncologists to help support studies in the variety of therapeutic areas. Finally, at Merit, we value collaborative partnership approaches, working closely with our sponsors and sites to build trust, partnership, and long-term relationships. In addition to the therapeutic area expertise and medical knowledge that the Merit team brings to our sponsors, our project management team brings a wealth of experience in a variety of study phases and designs. Our sponsors benefit from our Excelsior system, which offers a single, transparent, and easy-to-use application for the entire study process. Excelsior is programmed with a variety of response criteria, and the flexible system can be configured to manage many different study designs and accommodate all relevant modalities. Merit will actually be attending these 4 upcoming conferences in this fall and winter. We would love to see you there. Thank you all for joining us today. We will now open up the webinar for questions. Please feel free to submit them through the Q&A box, and we'll address as many as possible during this webinar. Wow, it looks like there's some questions that already came in. All right, well, got our first one.
All right, ready.
So, tackling variability head on. How is Merit specifically approaching reader variability in practice, and what mitigation strategies are actually being implemented today?
So Merit has a few different approaches. One of the first ones is disease-specific reader training, which is built and provided to readers before a study even starts. This can include things like disease and imaging background, in-depth discussions of tricky concepts like the pseudoprogression we talked about earlier for gliomas, and making sure we're keeping information and training up to date and current with the newest published literature and most recent developments for each disease. We also have something called a synchronization session, wherein our most experienced readers will walk the panel through real sample images together so that less experienced readers can see firsthand how an expert handles a disease's specific ambiguities and idiosyncrasies. Let's see. We also have performance monitoring throughout the trial. If there's a problem, outlier readers can be contacted for retraining mid-study, not just at kickoff. We also tend to have strong standardized BICR charters with criteria modifications, dating conventions, and missing data rules. All those things defined before the first patient is even scanned. Um, and all of this is important because how well a CRO calibrates readers upfront uh, can directly be reflected in the adjudication rate later. Let's see. Also, like we discussed earlier, it's important to favor quantitative over qualitative measurement where possible. It's not always possible, but where possible, it's ideal. Like I've said before, volumetric 3D imaging, where possible, such as in glioma, tends to beat 2D imaging, reduces variability, gives a better overall picture of the lesion, better accuracy. Um, PET tracers and other emerging more advanced technologies and imaging methods are increasingly providing higher precision and help to fine-tune the reading process. Also, like I said before, multi-component confirmation, so requiring progression call on two different criteria endpoints raises the rate of reader agreement significantly on like dates of progression calls of disease progression. Um, it's also important to give readers relevant clinical context ahead of time. Um, so discordance isn't driven by one reader simply knowing more than the other at the outset, um, which is called information asymmetry. Yeah, so I think that about covers it. None of these things can entirely eliminate variability because it's an
Mhm.
inherent part of blind and independent central review.
Mhm.
But these mitigation strategies can control the avoidable parts and can help contribute to ensuring a strong trial outcome.
Thank you for sharing that. That really does bring it all together. It looks like it looks like there's one or two more. So
Okay.
this one, can artificial intelligence read oncology images in this context? And more importantly, can it help readers reach higher agreement, not just replace them?
Um, the short answer would be yes, but AI is currently better positioned to act as a supporting tool, helping readers to agree rather than replacing them. Um, we can't have AI autonomously just making calls unsupervised. That would be a disaster.
Yes.
So AI already is being used in some narrow, already validated forms. Obviously, there's a lot more work that needs to be done, regulatory and QA wise. So let's see. For example, AI can assign RECIST categories directly from images or radiology reports, validate against reference standards. Um, like I mentioned before, automated longitudinal RANO tools, um, like the Brain Tumor Segmentation Challenge, have been aiming to produce um, software and AI tools that can produce objective, reproducible progression calls in brain tumors. There is some new evidence that AI can help improve agreement. Um, the European Congress of Radiology in 2026 had a multi-center reader study with AI-assisted CT scans, um, which reduced the reading time and improved consistency of tumor measurement across readers. There was also a bi-institutional study from 2024 where AI-assisted lesion segmentation was found to reduce inter-reader variability, and it also cut reading time by about 33%. For iRECIST, software-assisted assessment showed higher inter-reader agreement in one study than fully manual reads, especially at later follow-ups, which is right when discordance tends to creep in. But there's also been findings that there needs to be caution here, of course, because let's see, there was at least one study, an AI-assisted contouring study from 2025 that found that AI reduced variability overall, but also introduced important concept called automation bias. So readers were over trusting the AI output without the usual scrutiny, basically taking whatever the AI said as fact. And that's a common problem I think many of us have encountered already. It's a useful tool, but it can hallucinate sometimes. You have to double check it.
I
Um,
like that verbiage.
yeah, so human oversight and QA still matter for the foreseeable future. I can't really see that changing anytime soon. So the bottom line, I would say, again, as I said before, AI is a consistent assistant for the mechanical parts of reading, like segmentation, measurement, consistency checking, quantitative areas. And this can help free up human judgment for the genuinely ambiguous calls or the calls that require um, more perception or subjective interpretation. But again, I have to emphasize that it should be used as a supporting tool in helping readers to agree and not as a replacement to like an unsupervised automated reader. That would not be good.
I think we all can agree on that. Ooh, this last one I'm sure you can answer a few different ways, but would you say that this is a persistent problem or a solvable one? Is it an unavoidable byproduct, or could emerging technology and smarter mitigation kind of close the gap on that?
Yeah, so we already touched on this in the presentation.
Yeah.
Let's see if I can expand on it a little bit. Like I said in the presentation, about it's like 75% of discordant cases reflect just medically justifiable perception differences and not reader error. Um, so it's kind of inherent to BICR judgment, genuine judgment calls on ambiguous images for diseases with presentations can be difficult to read images for. That's difficult to avoid. Um, Again, the dominant source of variability is the BICR itself. It's kind of just built in. But intra-reader agreement can consistently beat inter-reader agreement and kind of offset it a little bit. Um, and again, some diseases like ovarian, glioma, lymphoma are inherently diffuse or poorly marginated, harder to measure the lesions or define them. And that's just biology. That's just nature. That's not really a process failure. That's just something that's unavoidable. It just has to be dealt with as part of the process. But there's a solvable part where we can close the gap and reduce variability rates. Let's see, like I discussed before, multimodal assessments such as labs, histopathology, and imaging in hematology in particular. Um, other measurement methods can help reduce the variability from just imaging alone. And then there's requiring a progression call on two or more separate response criteria instead of just one. That tends to increase reader agreement for when a disease is progressing. Um, New technologies like the PET tracers, more advanced technologies, increasingly providing higher precision, helps to fine tune the reading process. Volumetric imaging in gliomas, so 3D versus 2D, is much more precise and accurate and reduces variability from the readers. And then the AI assisted measurement segmentations is already showing measurable inter-reader agreement improvements in, in studies. So short answer, probably never zero. We won't ever get two human experts to agree on every hard call about ambiguous biology. But the gap isn't fixed either. It has already shrunk as criteria got more quantitative, as training gets more rigorous, AI tools matured, as we all learn in the clinical trial industry how to improve our processes. So the realistic goal is a small, well-characterized, controllable, residual level of variability. Um, can never really fully eliminate it, but that's exactly what well-designed trials with strong charters can already achieve.
Well, thank you so much for answering those questions. It looks like those three were the only ones we had time for today. But as a reminder, this will be sent out to everyone, so please feel free to revisit it again. And thank you, Will, again for sharing about this important topic. And
My
thank you
pleasure.
for joining.